So we'll get started right away. First speaker is Yousef Shafieyan from the HENS lab, and he'll speak on a novel cardiomyocyte-based device for cardiovascular drug testing.
Hi everybody, thanks to Ted Rogers Center for organizing such a great event and providing the opportunity for me to present my research work, in which I'm gonna talk about my project funded by Ted Rogers Center actually, about development of a device for high throughput drug screening using cardiomyocytes. So the question is, in spite of having numerous techniques in the market for the purpose of drug screening, what is the importance of having another device? I think the issue has been very well addressed by other presenters, including Dr. Mercola, Dr. Healy, and Dr. Radicic during their presentations. But specific to our case, basically, we don't have a comprehensive device right now in the market capable of measuring and quantifying the force amplitude of the signal and beating rate for cardiomyocytes in a high throughput manner. And we believe a device we originally developed for the purpose of fibroblast contraction studies can be a good solution for this issue as well, addressing these parameters in a high throughput manner. So I'm gonna walk you through my presentation showing you how we optimize the device for cardiomyocytes and how we calibrated it against a conventional method in the market, traction force microscopy for the force measurement, and how we transfer the idea from single dish to a multiple plate compatible with the high throughput screening technology. The field of two-dimensional contraction studies actually has been pioneered by the work of Harris and colleagues about 30 years ago, when they first introduced the very first version of the wrinkling substrate, which basically is a nano-thick polymer formed on a viscous silicone oil. And upon the cell attachment and contraction of the cells, these wrinkles would appear on the surface, and they can be quantified and be an indirect measurement for the force exerted by the cells. While it was the very first work in the field of visualization of live cell contraction, it came with its own drawbacks. Mainly, the surface was so fragile that it could easily rupture during the contraction of the cells and make it impossible for the further analysis of the cells. So over the past 3 decades, people have tried to improve the technique or introduce some other complementary techniques like traction force microscopy, which I'm going to talk about later. But the problem with all these techniques is that they are restricted to specialized labs, and they are not usually compatible with the high throughput screening technology. In the case of traction force microscopy, for example, it needs high resolution microscopy, which is not by any means compatible with high throughput screening. So we think our device basically, which comprises of a fully polymerized silicone-based polymer, can be a game changer, as it comes with features that can address the issue. I mean, by having the capability of measuring force amplitude of the signal and beating rate, as I'm going to show you later. And this is going to be an opportunity for introducing a device compatible with the high throughput screening technology. So one thing that I would like to highlight is that cells in vivo basically are experiencing the stiffness in a different range compared to what they would experience in vitro using tissue culture plate or gold-coated surfaces, which are very common for the purpose of drug testing. And the beauty of our device is that we can adjust the stiffness of the surface to come up with a range that more accurately resembles the stiffness that the cells would experience in vivo, including cardiomyocytes. And here you can see the images taken of a stem cell derived cardiomyocyte which were seeded on our substrate compared to the tissue culture plate, and you can see the difference when they are grown on our substrate with the stiffness in the same range as they would experience in vivo. These cells are generously provided to us by our collaborators from McEwen Center, Dr. Gordon Keller, and CCRM. So the way that our device works is basically by quantifying the wrinkles which would appear on the surface due to the contraction of the cell, in this case cardiomyocytes. But one could argue that the beating rate of cardiomyocytes, for example, can be also determined on the tissue culture plate, but just by following the contractile behavior of the cells and highlighting the area covered by these cells. But the signal that you would get from contraction of the cardiomyocytes on the tissue culture plate is so weak that you can barely see any changes over the time if there is a marginal change due to effect of any drug. So in the case of our device, basically the iCoD device, when we quantify the wrinkle area fraction due to the contraction of cardiomyocytes, this is what you can get from the device. And you can see the signal has been amplified in a significant way compared to what we notice from a tissue culture plate. So this means that we are able to quantify the cardiomyocytes behavior on our device in a consistent way by detecting the wrinkle area fraction. So the next step was to see if there is actually any relationship exists between the wrinkle area fraction due to the contraction of the cardiomyocytes and the force exerted by these cells. For that purpose, we use a technique, traction force microscopy, which is a quantifying technique for the purpose of force cell measurement of contractile cells. So the way that it works is that cells would be seated on a surface coated with the fluorescent markers, and due to the contraction of the cells, there's going to be a bead movement, and by detection of the change in the location of the markers due to the cell contraction, it's possible to come up with a deformation field map, as has been shown here by the vector map. And then by quantifying the wrinkle area coverage by thresholding the image, it's possible to compare these two parameters together. So for the purpose of vector map formation, basically we are using a fully contracted cell state and fully relaxed cell and compare these two together to come up with the idea how much beads have been moved due to the contraction of the cells, and we would have these vector maps. So we have done it more than a couple of times for at least for n equals to 11 so far, and we have shown that there is a good correlation exists between the average bead displacement and wrinkle area fraction due to the movement of these contractile cells. So what it means basically by knowing the area covered by the wrinkles, we can have a sense of indirect measurement for the force exerted by these cells to the surface. So the next step was to transfer the idea from single dish measurement to more compatible technology with the high throughput screening. For that purpose, we use glass cover slip and coated spin casted the polymer on the surface of the glass cover slip, and then mounted it to the bottomless 96 well plate, and use this 96 well plates for the purpose of drug testing experiments. Right now we are in the process of transferring the idea to 384 well format, as it's very identical to 96 well format in the sense of preparation steps. So to see if there is any compatibility with the high throughput screening imaging stations, we basically tested the sensitivity of our substrate to 3 conventionally existing technologies in the market for the purpose of high throughput screening imaging platform. And as you can see here, in phase contrast or bright field, it's possible to detect the wrinkles due to the contraction of fibroblasts in this case, meaning that our system is compatible with the existing high throughput screening imaging stations in the market. For the purpose of benchmark test, we had to come up with the idea of what we exactly we want to use as a benchmark test. And the state-of-the-art technology in the market right now is a device called Xcelligence, which is basically a gold-coated multiple plate. And when the cardiomyocytes are seated on the surface, they would basically start to contract, and due to the contraction of the cells, there's going to be an impedance change. And readout is basically the change in the impedance. And you can easily determine the beating rate of the cardiomyocytes using this device. But there are a couple of drawbacks with this system. Mainly, for this system to work perfectly, it needs cells beat in synchrony. And that is not the case, for example, when you are using cardiomyocytes, as I have had the experience. And another important thing is that the signal amplitude is not correlated with force using this device. So it's still the missing component. I mean, the force measurement is going to be something that we need to investigate after using this device. So we are planning to use this device at the same time for the benchmark test. And I have done some drug tests that I'm going to show you later. One of the concerns using stem cell-derived cardiomyocytes is that the cell population is not 100% pure. So it's very possible that we have fibroblasts, for example, in our cell population, and the concern is that does the signal from fibroblasts basically contribute to the entire signal that we would get from the contraction of cardiomyocytes? And we did this test. So basically, in the cardio region, this is the signal that you would get from the wrinkle area fraction changes over the time. And this is what you would get from the non-cardio region, in this case, fibroblasts. And you can see that signal contribution from the fibroblasts is almost negligible compared to what you would get from cardiomyocytes, meaning that from the entire image, what we would get is mostly due to the contraction of cardiomyocytes rather than any contamination from other contractile cell types in the cell culture system. So after optimizing and calibrating the entire system, we did some initial drug testing using our platform. And here you can see that we tested 4 commonly used drugs for the purpose of cell contraction studies. And these are basically the graphs that we would get from drug testing experiments. In the case of waving, for example, this is before drug administration, and this is after drug administration. And you can see changes in the signal are happening due to the effect of the drug, or nifedipine, bilibestatin, and isoproterenol all affected the cells in a way that the signal readout was different after drug administration. So I put everything together, and basically what we noticed was that the outcomes from our tests were in a good correlation with what we expected from the effect of these drugs on cardiomyocytes. To highlight one of them, for example, nifedipine is supposed to increase the frequency of the beating and decrease the amplitude of the signal. And this was the case when we did the measurements. And we tested the drugs at 3 different concentrations. We are planning to do more drug testing using other drugs and do the benchmark test with the cardiomyocytes coming commercially from the primary origin. So these are the very first results we have got from drug testing experiments. So in conclusion, we developed a platform for the purpose of drug screening using cardiomyocyte, using a device developed in our lab, and the results show that we are able to transfer it to an imaging system compatible with the high throughput screening. And the initial drug testing were promising, motivating us to go further steps. And for the future direction, we are hoping to do some more extensive drug testing. And at the same time, we are trying to improve the detection technique for the wrinkle isolation from the surface. So one of the things we can do is going for a more reliable fluorescence detection method. And it would give us also the opportunity to do further analysis like calcium signaling. So far what we have done is that using different fluorophores, in the case of rhodamine, for example, we have been able to detect the wrinkles when the cells are contracting and isolating the area covered by the wrinkles from the images taken over the time. So by the end of the presentation, I would like to thank my lab members from Dr. Hein's lab and our collaborators from Center for Commercialization of Regenerative Medicine and Dr. Gordon Keller's lab for providing us with the cardiomyocytes derived from stem cells. And I would like to thank the funding agency, especially Ted Rogers Center, for providing me the opportunity to work on this project. Thank you.
Thanks, Yousef. We have time for maybe one quick question. Yep.
Is this on? Yes. That's really beautiful work. It's very nice. When you polymerize this substratum, a couple of questions. How stable is it over time, and how flat is it from one position on, say, a multi-weld dish if you were to do that to another?
Yeah, so we have tested at least for 6 months. They are stable enough to do the test, and the surface is the flatness of the surface is consistent over the surface, because we are using a spin caster which just basically dispense the polymer over the glass cover slip, and the thickness is going to be something in the range of 25 to 30 micrometers over the entire surface.